You are listening audio paper series with Yasir, and we have Professor Mark Webber with us from Quadram Institute, Norwich, to discuss one of his publications, Triclosan Mechanism of Action and Resistance, published in Genome Research. Welcome, Mark, to audio paper series. And would you like to tell us what is triclosan and why you chose to study this?
Hi, Yasir. So, firstly, thank you for asking me to talk about this. So triclosan is a biocide, so it's an antimicrobial which has been studied quite a lot over the years, and we were interested in it for a couple of reasons. One, it's now really, really common in the environment at quite high levels, and there's been a lot of concern about the fact that it might select for resistant mutants, may also be cross-resistant to antibiotics. And secondly, its biology is interesting. So we know that at low concentrations, it stops cells growing. At high concentrations, it kills them. But that's not really well understood. So we really wanted to explore kind of how do cells respond to triclosan for those two reasons.
Okay, that's interesting. But then how did you investigate these questions in the study?
To see the impact of triclosan, we used a method called TRADIS Express. So this is where we have a very large pool of transposon insertion mutants in a bacterial strain, in this case E. coli. And we assay the role of all of them in survival in parallel. So we grow this big pool, expose them to triclosan, and then at the end of that, we identify all of the mutants that won and lost, if you like. So the ones that grew really well, the ones that grew really badly. And you can use a sequencing method to go back and map which of those mutants were more or less present in the pool. And the way we did it, we didn't just introduce transposon mutants that disrupted genes, but we also added a promoter into the transposon, which meant that we could also change the expression of all the genes. So in this one experiment, TRADIS Express, we can measure the role of all of the genes in terms of their knockouts, but also alterations to their expression in survival, in this case, in the presence of triclosan.
Okay, so in my understanding, in TRADIS Express, we can also study those genes that we cannot mutate with transposons that usually make up 7 to 10% of the whole genome. And it is possible due to the presence of outward-facing promoter. This seems a huge advantage over conventional transposon methods. But my question is, while you were studying this, you would have dozens of findings to share. But would you be able to tell us one or two major findings that are reported in this publication?
Sure. So we found lots of interesting things using TRADIS Express in this context, but there's a couple of particularly interesting things perhaps. So firstly, you're right that including the ability to change expression of genes means that you can assay those essential genes that you can't inactivate. And actually, we found that the method works really well for this. So we found a very, very strong signal for mutants that overexpressed a gene called FABI, which actually, as we know, is the target for triclosan, did very, very well in the presence of triclosan. So these mutants did really well, which confirms the method really works and that overexpressing genes can give you a very good signal. One of the other things that was really interesting is that we know that triclosan at low concentrations stops growth and at high concentrations kills. And we can see very clearly two different networks of genes involved in the response to either a low or a high concentration of triclosan. And that allows us to understand this kind of switch in its action but when it's at high or low concentration. So that was really nice to be able to see and document those genes.
Amazing. So you have studied triclosan using TRADIS Express, and you found the known targets of triclosan, as well as you found the bactericidal and bacteriostatic mechanisms. Do you think can we use TRADIS Express to study other situations apart from biocides?
Absolutely. So TRADIS Express is very versatile, and you could apply it to whatever condition you're interested in. Obviously, it has the advantage of altering gene expression as well as just inactivating genes. So yeah, it's broadly applicable to whatever condition you can think of. As long as there's a differential survival or growth ability for different mutants, then you'll be able to assay them using this technology.
Thank you, Mark, for introducing this study, and thank you all for listening.